Θα ήθελα να ξεκινήσουμε αυτή την ημερίδα που είναι αφιερωμένη στις δραστηριότητες της μονάδας Φαρμαπλή με έμφαση σε θέματα που αφορούν την COVID-19. Είμαι ο Γιάννης Σεμίρης, έχω εκλεγεί πρόεδρος στο Αθηνά και είμαι πολύ χαρούμενος που είναι ίσως η πρώτη μου δουλειά που έχω για το Αθηνά, παρόλο που δεν έχω διοριστεί ακόμη. Ξεκινάω να δουλεύω πριν την επίσημη ανακοίνωση, αλλά είναι ένας τομέας που με ενδιαφέρει και προσωπικά. Θα είναι μια ευκαιρία να δούμε τις δραστηριότητες της μονάδας Φαρμαπλή, αλλά όπως είπα δεν θα δούμε όλη τη μονάδα σήμερα. Θα δούμε κάποιες δραστηριότητες που είναι πολύ σύγχρονες και πολύ επίκαιρες. Η μονάδα Φαρμαπλή με συντονιστή τον κύριο Μαχέρα έχει εκφράσει το ενδιαφέρον της να κάνει και μια άλλη παρουσίαση προς το Ερευνητικό Κέντρο Αθηνά που θα καλύψει όλες τις δραστηριότητες της μονάδας και είναι κάτι που το δρομολογούμε. Απλώς αποφασίσαμε να μην το προγραμματίσουμε πολύ κοντά στη σημερινή ημερίδα, γιατί ήδη σήμερα θα μάθουμε πολλά πράγματα για τις δραστηριότητες αυτές. Λοιπόν, το πρόγραμμα υπάρχει στην ιστοσελίδα του κέντρου. Οι ομιλίες θα γίνουν στα αγγλικά, γιατί πιστεύω είναι καλύτερο για όλους και στην περίπτωση που κάποιος παρακολουθεί και είναι καλύτερα για αυτόν, αλλά και γιατί τα θέματα, οι ερευνητές εργάζονται σε αυτή τη γλώσσα. Οπότε προχωράω να παρουσιάσω τον πρώτο ομιλητή. Έχουμε μαζί μας τον κύριο Faidon Brotzakis, που εργάζεται στο Center of Misfolding Diseases, στο τμήμα χημείας στο Cambridge. Είναι πολύ ενδιαφέρον που μπόρεσε η οργανωτική ομάδα να φέρει και ανθρώπους έξω από το στενό οικοσύστημα του Αθηνά. Θα μας μιλήσει για την χρήση δεδομένων cryo-EM, κρyo-ηλεκτρομικροσκοπίας και προσομοιώσεις, μοριακές προσομοιώσεις στον καθορισμό της δομής και της δυναμικής όταν κατασκευάζουμε φάρμακα, αν καταλαβαίνω καλά, drug targeting proteins. Ωραία. Ο λόγος στον κύριο Μπροζάκη.
Καλησπέρα σε όλους. Ευχαριστώ πολύ την οργανωτική ομάδα για την πρόσκληση και θα προσπαθήσω να δώσω μια ενδελεχή παρουσίαση για το τι έχω κάνει το τελευταίο διάστημα στο Πανεπιστήμιο του Cambridge σε σχέση με την πρωτεΐνη του κορονοϊού, Spike, αλλά και ένα άλλο project, δεύτερο, που σχετίζεται με το Alzheimer. Οπότε τώρα το γυρνάω στα αγγλικά.
Ωραία.
Right, so let's see. Right, to begin with, I'm going to briefly talk about why we're interested in proteins and finding about the structures of their proteins they're the workhorses of our body. And as shown in this plot, they participate in several functional aspects of the cell. For instance, here, this is an ion channel. This is a membrane protein here, and ions transduce the membrane and thereby transduce the signal. Or, for instance, they catalyze enzymatic reactions. So this is proteins doing so. Or, for instance, when we're infected by a pathogen, then we create antibodies, which are, again, proteins, which are embedded in our cells, and they attack, basically, these pathogens. So, really, proteins are extremely important functional aspects of functional agents in our body, and really their function depends on their 3D structure. So, if we do know the 3D structure of a protein, for instance, we can decipher or envelope and pharmaceutical strategy where we target that protein in order to intervene into its function and thereby control the protein function on the molecular level. So this is main concept around which I'll be sort of playing during my presentation. And okay, so let's do a bit of a recap on what we know about 3D protein structures. So well, so here on the left-hand side, there's a table of different experimental techniques that identify the structure of proteins. Now, of course, this is the X-ray method. Is an extremely celebrated method that led to the discovery of, for instance, the double helix of the DNA and so on. And this was a method discovered here in Cambridge in the 1950s. There, when things went on during the 20th century, in the 1980s, we managed to get the NMR approach, which also not only gives an understanding on the structure of proteins, but also on the dynamics of proteins. And then this was followed by electron microscopy and so on. But I mean, it's kind of strange to think that although the proteome is in the length scale of millions of protein sequences, we only now understand how 150,000 of them look like. And the great well, of course, we do see a great rise in protein structures that we're now able to determine. For instance, look at the green curve, and also, well, basically the solid green curve is the crystallography ones. But now there is a prominent technique now that rose after the 2000s, with this electron microscopy method I'll be focusing on later on. But as I was saying, we only have a grip on 150,000 structures. So there is lots of work to do in this field. And I'm going to talk a bit about what challenges do this electron microscopy technique face, and how molecular simulations can augment or assist basically a structural determination problem, and thereby a function determination problem. Okay, so on the right-hand side, you can really appreciate these beautiful curves or, well, colorful images of proteins. Now, for instance, you can see on the top left, this is a ribosome, basically object or protein, where, well, translation or transcription thereby occurs. So each of these proteins, they have really a functional role, and they're extremely complex. But I'll go a bit about, in the next slide, what the challenges in protein structure determination are. And here I'm just, you know, showing a slide about an X-ray crystallography protocol, if you sort of speak, where you start with a single crystal of your compound, and then basically you observe a diffraction pattern. That is, after shining beams to your or electrons to your crystal, then you get some type of Fourier deconvolution, which you, with which you're able actually to trace an electron map, which you later on map into a structural model. The problem with X-ray and the limitation is that it's an extremely destructive technique, and this means that you really have to isolate your sample out of its biological relevant cell, for instance, where your protein acts into some plate and then crystallize it. So you completely, you know, destruct your compound. And then, of course, you have to turn it into a crystal and then determine a structure. Okay, but this is not what we would like. We would, and here is actually where the cryo-electron microscopy enters. This is a celebrated method that won actually the 2017 Chemistry Nobel Prize. And this is the amount of structures with increasing, the amount of structures without the years that have been resolved with an electron microscopy. And this actually curve grows at the moment. And the different blocks resemble the type of resolution one is able to get. And of course, well, we would like to have a resolution of, well, lower than 4 angstrom, and this is 10 to the minus 10 meters. So this is the resolution we would like to have. But of course, sometimes we get low resolution structures in this regime. And electron microscopy is able to address many types of length scales. It can, for instance, go to from millimeter And this is the electron tomography all the way down to the nanometer. And this is more like the cryo-electron microscopy that is able to access this type of length scales. But, whoa, sorry, what happened here? Oh, yeah, there we go. And this is like a very brief approach on why is cryo-electron microscopy able to, well, basically how it works. So basically, if you shine a beam of electrons through a frozen sample, so basically you extract your sample from some organism, from a cell, then you vitrify it. So basically you freeze the motion and the degrees of freedom. And in this sense, well, after shining light to it, you go through all different types of lenses that are able to magnify your signal to noise ratio, and then you're left with 2D images of your sample, of your protein, and then you have to reconstruct a 2D image to a 3D construct. And this basically plot here on the right shows how we're moving towards higher resolution proteins. So the main advantages, I mean, this was my introduction on this technique. So the main advantage is that it provides it gives access to a broad molecular weight range, so smaller or larger proteins, but also, and more importantly, can do that by basically taking samples that are near physiological, meaning that we don't really destruct the sample. And if you want to, I mean, in a different saying, or in a different way to put this, is that basically we really capture proteins in the act. Okay, now the problem with this approach is that it is not able to determine heterogeneous structures. So for instance, consider a structure that is a protein in a solution, and of course this is moving. So a protein exists in a native state, or in a functional state, or in a folded state, let's call it like that. But also often it undergoes conformational transitions, and it moves towards the unfolded state. Now, all of these structural ensemble, or these different conformations create sort of a superposition of different structures which do reflect the dynamics. But this poses a challenge for the technique, which actually then traces only a superposition of these structures. So if you do have a very dynamic protein, then cryo-EM faces its limits. Okay, and this is exactly the protein folding problem. So this is always the way I think about well, sort of structural biology or dynamical structural biology is that you, for instance, have a protein. So consider this protein, and also okay, so this is a reaction coordinate. Well, it's just a coordinate. It tells you how much your protein is folded or unfolded. So this is just a coordinate. And then this is the free energy, as we call it. So the folded state exists in the energy minimum. So this is the most stable state. But also a bit more infrequently, this protein can undergo conformational transitions and then exist in higher energy conformational states, which are less populated, but do very much do exist. So this is exactly, this is a free energy representation of a dynamic mechanism of folding. We go from an unfolded, low populated state towards a folded state. This is an extremely heterogeneous structural ensemble. And now we go to defining what a structural ensemble is. So a structural ensemble is not a single structure, of course. I mean, it's an ensemble. So you have different structures, as for instance, on the left-hand side, you have different conformations of the protein. And each of these conformation is associated with a particular, I mean, is a different state. We call it a microstate in physics. But then each of these states has a population, or a thermodynamic population, or a population that is basically depicted by the thermodynamics and the conditions of your system. Okay, so we do have structures, populations of each structure, and finally we do have interconversion rates, right? So we have a time scale to go from a state A to a state B and so forth. So all of these three ingredients constitute a structural ensemble. And the very fact that we do have fluctuations in the system, so we do the fact that we do have a different structure is of course reflected into the type of signal we absorb in, using any type of, an electron microscopy technique. So for instance, imagine that you have 10 states, each of them has an equal population, and each of them has the same interconversion timescale. Of course, the experiment would measure basically a superposition with equal weights of these 10 states. And then it would, so this is this is a very nice analog with a horse running. So if you really wait long enough, so you take a photograph and then you photograph a horse running, then you of course get a superposition of legs, in the front and in the back. But of course, this is not a horse, right? I mean, this is just a horse in different timescales taken together in the same moment. In any case, this very structural ensemble type of picture happens mostly in biology, because proteins actually, they're dynamic. They exist in a solution in high temperature. I mean, it's not in 0 Kelvin, thereby we do have motions. Okay, so, right. But of course, this very dynamics, as I was saying, it creates a problem into structural biologists, into decipher and into characterizing each of these structures and their populations. Very good. So I'm here just, putting forward some, very briefly, some experimental techniques. We talked already about X-ray, but of course there's NMR, SAX, FRET, and cryo-EM. I mean, these techniques are very nice, but of course they have the limitations, many of which I've mentioned just earlier. For instance, some of the challenges they face is that they're limited by, as any experimental or simulation technique, by random errors or by systematic errors. For instance, you can have also very sparse data in your experiment, that you have very few information about it. You can have ensembled average data, which is exactly the horse sort of photography example. You can have partial ensembles where you only have information on few states of your system. So all of these are extremely difficult problems when you are talking about structural biology, and these problems are kind of faced by experimentalists. So this is where basically we come in. So this was a long introduction, but I am hoping we're getting there. So, this is on the top row. You see the typical protocol for identifying a cryo-electron microscopy-based structure. So you start really from a vitrified 2D sample. So this is depicted here, where you can actually see the single particles. So this is cryo-EM is a single particle based method. It means that you, by viewing each particle in your grid, for instance here, this is each particle, that you can cluster it. So this is a 2D clustered particle-based approach. So this is a different particle than that one, and so on. And then based on this 2D classification, so each of these can, we can, using these 2D classes, we're able to reconstruct the 3D structure. Now, as I was saying earlier, if the protein is very heterogeneous or very dynamic, for instance, then your electron microscopy map, so this is the 3D electron microscopy map, and red means that you have very low resolution, meaning basically that you have very few electrons with respect to the volume, and thereby it means that the structure is not localized. So for instance, in these red regions, because the structure is not localized and because it's very dynamic, we're not able actually to associate a single structure. Now, if you ask basically, many editors in journals, they often really expect a single structure out of a single experiment. But of course, this is not realistic when you're talking about flexible and dynamic systems. So basically, for instance, this is the spike protein electron density map, and this is spike protein, I will be talking about it later on, is basically the protein with which the virus recognizes the host, or basically the cell, our body cells. Okay, so this electron density map doesn't really give you a unique structure. So basically, the structure that they constructed out of this 3D EM map had all of these very important red regions missing. So we knew nothing about these very important regions with which the virus binds to the host cell. All right, so as you will see later, but here I'm just introducing the concept, is that we can actually use molecular dynamic simulations in order to basically find out the structures of these missing regions. Okay, so what is molecular dynamics? Molecular dynamics is nothing but basically an atomistic representation of your system. So each atom or each element is one bead. And for instance, consider here this plot. This could be a nitrogen or a carbon, and their interaction is given by some type of a bonded interaction that we model by a harmonic. And then, of course, we have interactions about the rotation angles between atoms, and these are again harmonic and so on. And then we can quantify also the electrostatics interactions between different atoms. And all of these type of interactions are modeled into a potential energy function that, well, basically is given, it looks like this one. And of course, as anything, any model has errors. So of course, and basically the very reason we're combining molecular dynamics with experimental data is in order to basically sort of basically move forward and go beyond the errors of molecular simulations or electron microscopy, as mentioned earlier. So in the end of the day, we're sampling configurations or structures that come out of a physics-based approach, plus a likelihood that biases our distribution of structures that comes out of the molecular dynamics. So it biases it also, or if you want, it modifies it with experimental information. So in the end of the day, we're sampling a distribution of structures of the full structural ensemble. Now we are not, we can simulate the full spike structure. And basically these distribution of structures obeys both a bottom-up approach, which is a physics-based approach, simultaneously with a top-down way, which is basically the cryo-electron microscopy data. So this is basically the structural ensemble. Okay, I'll be talking about it a bit more briefly as we move forward, but in the end of the day, and before anyone asks, so a main issue, so this is how the energy function of the meta-inference approach looks like. So we do have an energy. So consider that you have a molecular system that sort of propagates in time. Then the way that this motion propagates is based on the energy of the physics-based approach, which is called molecular dynamics. And then this energy is penalized by an amount which is quantified here, and this is the deviation of your configuration with respect to the data, and this is the EM data. Now, of course, the EM data do have an error, so the EMI method is able to quantify also the error in the data. So for instance, if you have data like electron density data that are extremely erroneous, so the sigma here is very big, then the restraint is very small. So in other words, we don't trust much the data if they're erroneous. So this is the energy function of the EMI. Fine. That's fantastic. So that I hope that we can now jump into the applications. Okay. So first of all, I'm going to talk a bit about something that is very related to the Center of Misfolding Diseases in the Department of Chemistry, and this has to do with a structural ensemble. Well, basically, so we determine a structural ensemble of a complex, and then we decide how to modify or mutate amino acids in order to, well, improve a function. But I mean, this will become a bit more obvious as I'm going to this slide. So, fine. So this is this is one of the hypothetical, a hypothesis mechanism of how one could be left, I mean, how the Alzheimer's disease propagates in the brain. So consider a neuron cell here, and consider basically, right, so this is the microtubules. Okay, so the microtubules are, it's just a polymer, so basically, it's a polymer of tubulin units that is stabilized by a protein called tau. Now, microtubules are very important. So what they do is they polymerize and they depolymerize, and they really contribute into the signal transduction along the neurons. So this is a very important function. Moreover, there are some cargo proteins that walk through on top of these polymers or fibers and carry on other proteins. Anyway, so microtubules are very important in memory, signal transduction, and so on. There's a protein called tau, which is an intrinsically disordered protein, meaning that it doesn't really have a stable state form. I mean, a folded stable state form. Anyway, so tau, what it does is that it binds to these microtubules and it stabilizes them. So it really promotes the assembly of these agents. So in other words, this is what tau does in a functional form. Okay, now the phosphorylation hypothesis is that you can have modifications of some amino acids at the surface of tau. For instance, serine turns into a phosphoserine and so on. And because of this aberrant hyperphosphorylation of tau, then tau loses affinity from the microtubules, it enters the solution, and thereby it bundles with itself, forming these neurofibril tangles, which are actually pathological. So if you open up a brain of an Alzheimer's disease patient, you often find the consequences of these fibrils or of these oligomers, which basically disrupt completely the membranes of the neuronal cells. So this is what it looks like, more of like a Swiss cheese. Anyway, so this is an artifact or a consequence of the fact that this hyperphosphorylation occurs, and then tau leaves to the solution, and it polymerizes. Fine. So what we really wanted to do is, so what we really needed to do is to have a protocol that is able to identify sites at the surface of tau that are phosphorylation prone, so that are more likely to be phosphorylated, and at the same time, if they do get phosphorylated, to find out whether actually their function or their stabilization capacity changes. Okay, so to do that, we do again need a structural understanding of this type of interaction. So this is the sequence of tau, it's a long protein. Anyway, so we simulate a 200 residue of tau on top of the tubulin unit. And in yellow, we show this is tau, and then the microtubules or tubulin units are in green and blue. And I'm showing here in red what was previously characterized in this complex. Now, the reason that only that small segment in red was characterized was that, I mean, people were not able to assign a single structure into the EM map. So what I want to say is that, the electron microscopy gives you this type of map. So if you take, so the first map on the left shows high electron density regions. And of course, you are only able to assign a structure using the high electron density map. So this structure in red was characterized by using, the information of high electron density. But I mean, there is much more richness into this map. So you can have a low electron densities here, where you are not able to assign a single structure, but I mean, they clearly depict that you have disordered C termini in the tubulin and so on. That's fantastic. So we were very happy to really simulate the entire structural ensemble of the tau and the microtubule for the first time. But then we wanted to ask some meaningful questions. So, for instance, We quantify the interactions of particular amino acids. So this is the amino acids, the serines of tau. So how many contacts these amino acids form, and how flexible these amino acids are. So, for instance, you can appreciate by looking seeing that, for instance, there are some amino acids like serine 262 that forms many contacts with the microtubule, and contributes much to the stability. Now, if this serine is actually phosphorylated, as it does happen in the Alzheimer's disease, well, basically you do get these tangles. So this was a very nice check to really see that, that high affinity serine residues as quantified by structural ensemble do show an Alzheimer's disease type of propensity once phosphorylated. So we really like that fact. And then you can really actually have a structural insight on how the serine here in red interacts with the glutamic acid in low populated state. So this is in a ground state, so this is a high populated state or a very low energy. And this is more of a low populated state, which is where the serine is solvent accessible and their kinase can phosphorylate it and so on. So this is, but of course, to verify that now our protocol, so basically our serine here, which forms lots of contact, is a very important residue that would increase the Alzheimer propensity. We really had to verify that hypothesis. And then what we did is that here, Phil and Ryan, they did a microtubule assembly, polymerization assembly assay, where you phosphorylate that particular serine, and then you map how the polymerization slows down. So as you probably remember earlier on, I said that well, if you phosphorylate here and you lose affinity, then of course the microtubules can't really polymerize anymore, because actually tau catalyzes the polymerization. So this was really assuring as to the fact that this serine actually and its phosphorylation really relates to the affinity and to the stabilization of these microtubules. We did the same thing. We screened actually now so we also screened for the acetylation event, that is for lysines and how strongly they interact with the microtubules. This is a very similar approach, and we identify this serine 311, and indeed actually once acetylated, there is an effect on the microtubule assembly. That's very good, and this is extremely it was a nice result and pretty assuring that our structure-based approach on the functional role of tau and the effect of post-translational modifications protocol actually works. And this is me concluding the first part. So yeah, so we were very happy to have a structure-based protocol to predict and test regulatory post-translational modification on tau microtubule complex. Now I'm entering my final and I think, well, more related topic of discussion, and it has to do with the structural ensemble identification of the open to close mechanism for spike SARS-CoV-2 for in silico drug discovery. Okay, so this is us today with respect to the pandemic. So this is the confirmed COVID-19 deaths as a function of, well, time. And it is very obvious that this pandemic actually undergoes in waves. And so this is extremely, we are really in the midst of a hurricane, and we need approaches, like bottom-up approaches or top-down as well approaches, or different types of ecosystems in order to address this pandemic and in general the emerging pathogens in the future. So from my point of view, well, as a simulator, I think that there is a great into the great increase in computational power. So for instance, here you see a GPU or a cluster or a supercomputer. So the amount of basically power one can have, but also distributed computing is now pushing the boundary of all of these computer-based type of approaches. And here, actually, more intriguingly, you might realize this is a plot where we show as a function of time. This is the usability, or how many users basically use, well, the amount of users having used a distributed platform called Folding at Home. Now, Folding at Home is basically a platform, a distributed computed platform where you borrow some small percentage of your CPU into a project, and the particular project of this is the Bowman and the Codera team. Their project there was to simulate long enough these different proteins of the coronavirus. And this is actually an extremely nice approach into a citizens-based sort of supercomputer. And I mean, as you show here, so this is the amount of places the users donated their computing time. And I mean, this is extremely intriguing, the fact that the computer that was created by these donators for this particular COVID-19 simulation case actually exceeded, well-known state-of-the-art top supercomputers like the IBM Summit and so on. My point being here that there are various ways to utilize at this moment during the pandemic the computer power, and there's a great sort of offer and actually demand on it. Let me dive into my project. So so consider this viral envelope where you have a capsid in green, and then you have the spike proteins in red. And this is basically the entire virion. All right, so spike basically recognizes a particular, so this is the first step of recognizing a particular receptor in the cell. So this is the ACE2 receptor. So once it recognizes it, so this is the first step, the recognition, then it goes towards the second transition, which is the so-called fusion transition. So then the membrane of the virion opens up, and then the genome, so the RNA of the virus, enters the cell, and thereby, basically uses the machinery of the cell to replicate the RNA and so on. Okay, but to focus a bit more on this particular part, so this is the recognition step of the spike with the ACE2 receptor, because this is the very first step of the attack. There you have spike on the top here. So this is spike here, and you have the ACE2 receptor, fine. But I mean, there are two states, and this is the so-called conformational masking strategy of spike, where spike can be in the open state. So this means that this RBD region is open, facing the ACE2 receptor, and this is more of an active state. So this means in the open state, spike can bind the ACE2 receptor, while in the closed state, the RBD is pointing inwards, and then it can't really bind and recognize the ACE2 receptor. Now, this type of conformational masking strategy is an extremely smart way for the virus, so for the spike to defend from antibodies, because I mean, if you only had an open state or an active state, then many more antibodies would be able actually to attach to spike, and thereby begin an immune response process that would probably evolutionarily minimize the outcome of the COVID. Very good. So so our approach, okay, so as I told you in the very beginning, basically the structure of the spike identified by the cryo-electron microscopy had key regions indicated in red here missing. I mean, there is extremely dynamic, that's why they're missing. But of course, by applying our approach, we're able to bring to light all the different type of open and closed conformations of the spike. By the way, note here that this blue brush, if you wish, are basically glycans. So these are sugars that that many viruses use in order to protect or shield themselves again from neutralizing antibodies and so on. In any case, we're able to define a structure, to derive or to determine a structural ensemble, which looks like this. And on the right, this is a movie of the different type of conformations existing in this ensemble. All of these conformations constitute to the very low electron density of these dynamic regions, by the way. And also, if you want to, when we're trying to map down the different structures on a type of a landscape or of a map, so this is the XY axis. So the X-axis is the distance from the closed state. So the closed state is here, and the Y-axis is the distance from the open state. So open state free energy minimum exists here. So this is this state. But then, of course, there is an ensemble of structures and a pathway towards going to the closed state. So in fact, there are actually two pathways. Very good. So, well, after, of course, understanding this complex rearrangement of the RBDs and so on, I mean, the question was, what can we do about it? And this is all, by the way, in a preprint we have in Kerm Archive. So we really thought what we can do with this. So we really have free energy minima, some of them are less populated and so on. So basically what we do find is that there is a state, sorry for the back and forth here. So there is this state called C2, which basically is able to, this is the state C2, where there is a void space between the RBD and the N-terminal domain, where
Faidon,
potentially, yes.
Excuse me to interrupt. This is very interesting, but are we getting close to the wrap up?
Yeah.
Because time is pressing.
Yeah, we have 1 minute to go.
Good.
Okay. Yeah, sorry about this. So basically, we're identifying a pocket where a drug, a small molecule can bind to, and we are actually screening then a library of small molecules that would potentially enter this higher energy or low populated state. And we do find that there is indeed a compound that binds well there, that binds strongly in the cryptic pocket, and also in the same time reduces the viral load. Now we actually have to perform experiments to see whether the reduction of the viral load of that particular compound is because of the binding to spike or the intervention to a different level on the virus. Okay, so this is the conclusion here, that we do now have a structural ensemble and atomic understanding, and that we have identified a binding site in a small molecule that can potentially regulate or jam the spike RBD dynamics, because we really want to jam this open to closing transition by introducing a small molecule to some cavity, so that the virus probably remains in the open state, so that it is exposed to neutralizing antibodies, or only in the closed state where it's actually not inactive. Fine. So to finish with this lecture, I think, so this is the typical pipeline on if you really want to make a vaccine, or you really have to select an antigen region. So the way we I think see the structural ensemble is not only identifying small molecules, but also having this structural ensemble I showed earlier. We're able to find sites on the spike that are most vulnerable, actually. So these are called epitopes of the antigen, and this holistic understanding of epitopes would probably lead to more effective vaccines, or can lead to structure-based targets for neutralizing antibodies. And to wrap up, this is my final slide on, I really think that COVID research is extremely multidisciplinary. It needs lots of input from different aspects to maintain social balance. I mean, good epidemiological data, accurate testing tools, primary care testing, and vaccine centers. But of course, efficient vaccines that are probably led by also computer aspects, as I showed earlier. So I want to thank my collaborators, Michele, Thomas, and that have been associated with the first work. I mean, this is Michele Vandruscolo, my supervisor, but of course, Phil, Ryan, and Gonzalo, and Max, and of course my friend Kostas for advice on the immunological aspects of the project. So of course FEBS for funding, and the Center of Metabolic Disease, and the university, and you for your attention. Thank you.
Thank you, Faidon. Thank you very much. That was very interesting. We have some time for questions. As promised, it's a bit shifted, but I'll take a couple of minutes from the next session for questions, please.
Can I please ask?
Sure.
Yes.
Could I have the... Okay. Dr. Brotzakis, first of all, many thanks to Athena researchers for this event. But it was a major advancement concerning AlphaFold, which uses a novel AI technique in simulations, and it was developed with DeepMind, the interaction between cryo-EM and computational. I mean, does it take us a step further in successful? Take time to show. Thank you.
I think it was a bit, well, okay, so if I understood well the question, because I didn't fully heard well, so AlphaFold is an AI-based approach to determine the 3D structures that is extremely recent and does extremely well for the ones that it has been trained for. So it really depends whether we do have good representation of membrane proteins in the PDB database, so on how well that would do to determine the ground state structure of the protein. But even if we did have data on it, I think that it would only determine the ground state structures, and thereby, you know, the dynamics, which is very important, would still be an open question. As to the algorithm per se, and last time I checked, the data, or I mean the Python notebooks and so on to apply it were not accessible. But I do really think that the AI and how basically it enters into learning the code and the 3D, well, basically interactions, I think is extremely important. Not so much, I don't really see that you will automatically get an insight into the dynamics, but I do see how much it can help into identifying well this chaotic type of interactions that protein, you know, form.
Let me complement that comment. I think AI would be very useful also in studying cryo-EM data. In fact, it's something that we are studying these days in my team, how to go from these maps, from the cryo-EM maps to the 3D structure. There has been a few papers the past year that apply machine learning. Of course, they face the problems that you mentioned, that we get a partial structure. So I don't know if you had a comment on that, on what exactly are we looking in when we obtain this partial structure. So in my case, it's machine learning, but there are many methods. And then you would like to apply simulation to get the complete structure, the full structure. Are there any characteristics that we would like to emphasize in that first step? I think this could be of interest to many researchers that are listening. So just a short comment, please.
Yeah, so
yes, exactly there.
Yeah, in order to go from the top, from this map to basically this ensemble, you really have to encode basically the information, so the 3D structure information. For instance, one way to do it is to generate, you know, use for instance a generative model, which basically is then creates, of course, models that are penalized based on such a, I mean, it can be much easier, based on a scoring function that quantifies the interactions. So I think this is a good way to go, for instance, with a GNN, a Gaussian, a generative neural network. This would be a nice approach. Yeah.
Yes. Okay, good. That's a good idea. More questions. I think there is at least one.
Yes, if I may, first of all, congratulate you about a very nice presentation, very clear. Just one question, or actually, if you could elaborate a little bit on the identification of the post-translational modification binding sites that you mentioned.
Yeah.
How exactly did you identify those sites, first of all? And regarding the flexibility that you mentioned, whether you have taken into account the solvent contribution?
Yeah, so first of all, the simulations are always done with explicit solvent. So when we're talking about, so that's point one. Now, what we did is we say, okay, look, we want to find which, say, phosphorylation sites contribute to the, or basically destabilize the complex. That's the question. Okay, which phosphorylation sites destabilize the complex? Fine. So then we take all serines, because serines can get phosphorylated. So this is all serines and some threonines, all threonines. And then we quantify how many contacts serines and threonines form. So how many contacts the tau serines and threonines form with the complex. This is the number of contacts. For instance, on average, this serine forms many contacts with the microtubule. So since this serine forms many contacts with microtubules, one can hypothesize that if you modify that serine to a phosphoserine, then these contacts might be lost. So this is an important, actually, so high contact amino acids are very important for the affinity, and thereby we filter these high contact amino acids. We take them, we phosphorylate them by chemical mutagenesis approach, and then we check if that phosphorylated tau binds well to the microtubule. Now, it doesn't bind well to the microtubule because the microtubule actually forms much later. And as you saw earlier here, so the microtubules are only form when you have tau attached to it. So if the microtubule doesn't form, then probably means that tau doesn't attach to it. So this is a delay in the formation of the microtubule, thereby a loss in tau attachment to the microtubule. And that loss is associated with the phosphorylation of the serine, which this serine we decided to phosphorylate because it contributes much contacts to the microtubule. That's the thinking. So we take these right-hand side candidates, and it's quite interesting the fact that you do see that, of course, it's not a one-to-one, but these yellow cases, so the Alzheimer's disease phosphorylated serines are really contributing more interactions compared to the healthy, so phosphorylations happening in healthy brains. So these are really not regulatorily amino acids undergoing PTMs.
Thank you.
Okay, maybe one last brief question. Okay, good. Then we'll thank you again. I think it was a pretty dense talk for most of us because we have a different background, but we are very eager to work on such things. And I think there are many computational issues. I don't know if it was clear for everyone, but for people that have some experience, it seems there is a lot of computational issues. So thanks again. Let's move on to the second session today. Okay, I will introduce Professor Tzoulaki while she's sharing her screen. So we are very glad to have with us Ioanna Tzoulaki, who is working, she's faculty at the School of Medicine at the Ioannina, and she'll be talking about modeling the COVID-19 outcomes. I believe this is another very relevant aspect of what we are doing in Athena, in the sense that we already saw some technical issues before, but I think risk factors and risk prediction in modeling is something that we may have seen, but maybe in different contexts. So we're looking forward to that. Professor Tzoulaki, the floor is yours.
Thank you. Good afternoon to all, and thank you for the invitation. It's a fascinating day with diverse talks. We are going to change a scene here, going from the micro level to the macro level, where here we are not looking at at the data concerning the microstructures, but we have population health data. and how we can use this data to make either predictions of whether someone is infected or will be infected, or on the outcomes of these predictions. So starting with, let's see. First of all, a few words on why we need this prediction model, and introduction in case some of us are not familiar with the literature. Most of the times, healthcare providers are facing critical time to make sensitive decisions regarding treatment, regarding triage. So we need decision support tools. A lot of the times, these decisions are made inherently, in our brains. But if we have mathematical algorithms, they can help us making better decisions across different settings. So decision support tools are commonly underpinned by what we call clinical prediction models, which are multivariable models that use a lot of variables weighted according to their importance to calculate risk. These models use patient data and calculate the predicted probability. That could be either that something exists now, which are diagnostic models, or whether someone has a condition, or future outcomes, which is a prognostic models that have to do either with disease deterioration or disease onset. So in terms of going to COVID-19 now, there is an interest in prediction models of COVID-19, either, first of all, because we know that the risk of complications varies. So it's different according to patient characteristics. For example, we know that elderly patients, persons with comorbidity are more vulnerable to more severe morbidity and mortality after infection. And because this information is very critical, in order to make treatment decisions and care, in order to triage patients when allocated limited healthcare resources. So if we know who is more at risk of severe outcomes and mortality, we might be able to triage those that go to ICU, for example, or to estimate the burden of the healthcare system, and even to prioritize vaccines in order to give the vaccines to those that are at higher risk. So there is a scope for use of those risk models. However, I should say that since I'm working in the area of prediction research, that we have failed in different settings to find prediction models that can be used in clinical practice. The literature is full of these models, but we have very few success stories. So one could be the cardiovascular risk algorithm, such as the Framingham risk score and the Euro score, based on which we initiate preventive treatment with statins. Or it could be models that we use to calculate the risk of Down syndrome during the ultrasound. Or there are very few examples that we use in clinical practice, but there is a lot of research interest and a lot of papers on this setting. So when the pandemic initiated, we realized that there are plenty of prediction models populated in the literature, and we were interested in that. Before going to what we actually did to gather this evidence, we started thinking at what do we really want to predict for COVID-19 and SARS-CoV-2, because there are different elements in all this and throughout the research of risk of COVID-19. So first of all, there is a risk of infection. So whether we are studying effectivity, whether someone, given that they are exposed, will be infected and will develop symptoms. Then we have the probability of being infected when having symptoms, so having a symptomatic infection. So one is whether you will be infected, then whether you will develop symptoms, and then whether being infected, you will develop severe symptoms, going to hospitalization, death, or other adverse outcomes. And all these different outcomes need different study designs and different populations. So it's important to have in mind what exactly we are trying to answer each time. And then prediction research has its own special methodological characteristics, which are different to common epidemiological analysis that we commonly use to study etiology. So here we are studying prediction, we are not interested in etiology, and we have special methodological methods that we are interested in. And we have things like validation and calibration and different metrics that will enable us to study better prediction research. So having all that in mind, we started this systematic review that is published in BMJ. And systematic review, you will be familiar, it's the systematic gather of the evidence across a theme. So our aim was to gather all prediction models for COVID-19 and to critically appraise them. So not only to gather them, extract important information and also calculate the risk of bias, whether they are good models, in order to select some that could be implemented in clinical practice. That was our aim. And this is a living systematic review, which means because we were aware that the literature changes in real time, we agreed with BMJ that this will be a living systematic review, so it will be updated every few months. So every few months a new paper is uploaded on the website of BMJ and then published, because this is a topic that changes very much, and new papers are published. Now that was a huge undertaking because of the number of studies that are published in the topic, which are a lot, and the fact that a lot of them, the vast majority of them, are poor quality. And when a study was of poor quality, it was even more difficult to extract the information and to assess the risk of bias, because it was this information was either not there or it was hidden somewhere. So what were our eligibility criteria? Which studies were we interested in? So we were very open. So we included studies if they developed or validated a multivariable model or a scoring system based on individual participant data to predict any COVID-19 related outcome. So we included diagnostic models to predict the presence or severity of COVID-19 in patients with suspected infection. And we created prognostic models to predict the course of infection, and prediction models to identify people in the general population at risk of COVID-19 infection. I have to say that this last category was the one, we didn't have a lot of models there. Most of the models were diagnostic or prognostic among those that had the disease. And we used the standard PubMed systematic review methodology, identified papers in PubMed and preprint articles. Later on, we stopped having preprint articles at the beginning because most of the articles were on preprints. We were doing the peer review ourselves, but then realizing that, a lot of those are moving into the literature, peer review literature space, we stopped that. We used an artificial intelligence text analysis tool in order to improve the sensitivity of the algorithm and select studies that were likely to be eligible. And then we had two reviewers that reviewed those articles selected by the tool and select those that were eligible. And then we use standard checklists for systematic reviews to extract the information. So to do the data extraction per article, that was done by two independent reviewers. And when there was inconsistency, a third one resolved the issues. Now, what did we find? This is the flow chart of the studies that we selected. You see that overall we had 169 studies. We reviewed more than 37,000 records. And of those studies, a lot of those studies presented more than one model. So we had 232 models. Of those, 208 were unique. And dividing those, I told you before that the models to identify people at risk in the general population were uncommon. But we had a lot of diagnostic studies. So studies trying to diagnose the risk of having a COVID-19 infection. That was very, there was a lot of interest on this at the beginning, where we didn't have a lot of, we didn't have the rapid test, the PCR tests were very constrained, we didn't have a lot of PCR tests, so there were a lot of research groups trying to predict algorithm based on images and CT scans to predict whether it was COVID-19 or something else. I think now the the interest in this space is decreasing. And then we had the prognostic studies, which I think this is where the interest, our interest is, and it's the most interesting space, where we have prognostic models to predict mortality or progression to severe disease or critical state. Now, of those, only a small proportion provided enough information on the equation and the intercept of the equation in order to allow us to recalculate this model if we wanted to. So the reporting was poor in that concern. Some of them presented a tool that others could be used, so it would be an online website or something like that that could be reused. 13 of those had a web calculator. Now, moving on some characteristics of these studies, a lot of those were on single country, so they were population from a single country, which is understandable given the data that we had at that time. Most were in ages, elderly populations or middle age. We had only one model in pediatric populations as well. Now, among the diagnostic studies, those reported the prevalence of COVID-19 varied between 7 to 71%, and that's, it is interesting to see with how different populations these diagnostic studies started, and the same with the prognostic models, where a lot of studies develop prognostic models to predict mortality risk and the percentage of death rates from 1% to 52%. So they were having either very high risk populations or very low risk populations. Here we see the sample sizes are not great either. And we see that, the breakdown of prognostic and diagnostic that we saw before. These are only the 7 models that predicted COVID-19 in the general population. I think that there is not a lot of interest here. It's not clear use of these models, so that's why we only had a few. Moving on to the diagnostic models, we had more here. Those models were trying to distinguish between patients with and without COVID-19. With C-index, the C-index is a discrimination matrix, is relative to the ROC curve, the AUC, but using survival data, range between 0.65 to 0.99. So anything above 50 means that the model cannot discriminate, and 1 means the model perfectly discriminates. We had a range of predictive ability of the models, some of them very close to being a perfect model, which is hard to believe, because the models we have in clinical practice, the best of them are 0.75 or 0.80. So we haven't really seen a model with 99. It makes you wonder how they managed to reach as high predictive ability, and whether that's, overfitting, and whether if you validate it in an independent set in a different population, that discrimination ability will deteriorate. So 75 models, we are talking about diagnostic models here, were proposed to support the diagnosis based on images. So we had a lot of papers on images using mainly deep learning techniques. And the problem was that a lot of those papers, they all used the same set of images that were uploaded from China at the early ages of the pandemic. So they were all using the same set of images with the same limitations, and reporting great models of excellent discrimination, but they were so biased, the images and the control populations, that it was obvious that these models were with high risk of bias, and we will discuss that later on.
Excuse me, these are images of what?
They were CT images of the chest, And based on that, so they had cases and controls, and based on that, they were predicting whether it is, pneumonia from COVID-19 or from something else, from flu, for example. Yeah. Now, these are prognostic models. The title here is incorrect. So these are the prognostic models, where we have 107 prognostic models. The intention to use this model was unclear. So it wasn't clear what would be the use of this model. Would it be for triage, for treatment, for other kind of prioritization? And also the prediction of horizon, which means, how you're predicting outcomes in a few days, in, a month, later on. It was unclear. Most of the times it was not reported. And it varied from 1 day to 37 days. 39 estimated mortality risk, and 28 aimed to predict progression to a severe or critical disease state. And the most frequent predictors, those that most models had, were, age, the usual comorbidities that we know that affect COVID-19 outcomes, either vital signs, images features that usually it's CT images of the chest, male sex, and biomarkers like lymphocyte count and C-reactive protein. And again, the C-index here was good most of the times, from 0.68 to 0.98. Now here is the risk of bias assessment. So this is a common tool that Cochrane also uses to appraise the risk of bias, where green means low risk of bias, yellow is unclear, and red is high. And you see here the overall, all studies, general population studies, diagnostic studies, separately for images, and prognostic studies. And what we can see across all those is that, we see a lot of reds. We have high risk of bias overall, and that is coming mainly from the analysis part. So this is where we had the major limitations on the studies. Also, in terms of selecting the participants, we had bias samples on how to define the outcomes as well. If we looked at the more in detail, I would say that in diagnostic models, the major issue here was that let me find this. Yes, in diagnostic models, we had a lot of unclear study design of how this data was collected, and how these controls were selected, whether the controls were coming from the same population or there were very different controls, so we were making unfair comparisons. The definition of the outcomes were often not defined or defined in different ways in participants. Now, in terms of prognostic models, the common pitfalls had to do with dichotomization of predictors, where we lose a lot of information, unclear inclusion and exclusion of study participants. So a lot of the time we had prognostic models within populations at the hospitals, where the people that remained in the hospital and the study had ended had an unclear outcome. So they didn't develop COVID-19 and they weren't discharged from the hospitals. And a lot of times these participants were excluded from the studies. That was a big bias introduced there, because a lot of these patients hadn't developed severe outcomes yet. That's why they remained in the hospital. So there was a bias introduced by this. And when the analysis was done, the censoring was not taken care, or the competing risk analysis. Also the calibration of the models, how well the expected, the predicted probabilities and the actual probabilities match was not assessed. And also we had very few studies having validation, which was very important for these studies, especially the size, the sample size was small. We didn't have external validation. Only few of them had validation of their models. Nonetheless, we identified two models that we thought they may be good, or at least they had an unclear risk of bias. One of them was diagnostic, and one of them was prognostic. So the diagnostic one presented findings from developing a diagnostic model, but had substantial missing data, and it remained unclear whether the use of their, they used median imputation for the results, which is an unclear strategy to deal with missing data. And then we had Knight et al. That they develop a prognostic model for in-hospital mortality. However, we did have, again, dichotomization of continuous predictors, what else did we have here? I think it was unclear reporting overall that made us have an unclear here. Nonetheless, it was selected as one of all those 107 models that had probably the best reporting and probably the best model we could have. Now, later on, in summary, we saw that all models reported moderate to excellent predictive performance, but all were appraised to have high or uncertain risk of bias owing to a combination of poor reporting and poor methodology. And out of all this model, only 46 carried out an external validation, and calibration was even more infrequently used. So we cannot yet recommend any of the identified prediction models for widespread use in clinical practice. Now, from the time that the last update was published, we had the publication of another model, which is the Q-COVID model in the British Medical Journal. This model uses a large proportion of the electronic healthcare data in the primary care records of the UK. And it used the state-of-the-art analysis and reporting. So here we had enough sample size to develop the model, to validate it internally, to present calibration analysis and variable selection approaches that were valid. And that model was used by the UK in order to identify people that need to shield, so those that need to protect themselves. It's on the website of NHS, where you click on the Q-COVID and you calculate your risk based on that. It has factors like age, sex, the year of birth, ethnicity, BMI, and comorbidities. So nothing, we didn't expect as a risk factor, but a combination of factors and weights that considered appropriate. And this model was later used in order to prioritize these people for vaccination in the UK. So people that are considered high risk based on the Q-COVID are now being prioritized for vaccination. So we see an example of a model that is implemented now in clinical practice using, big data and linkage of primary care records and electronic health records. Moving on, that's one of the study I thought of discussing here with this group. Excuse me, within this same space, I wanted to present this smaller study in order to make us think again of how difficult it is to answer questions in terms of disease risk with population health data, where we have some people that have been tested for COVID-19, but these are always very biased samples. So it's very difficult for us to answer questions in terms of, disease risk and severity risk. So we have an interest to examine risk factors for developing COVID-19, but population health data poses special challenges, and I'm going to give you an example of that. Because a lot of times it's difficult to distinguish whether we are examining risk factors associating with infectivity, so whether you are infected, or with developing symptomatic disease. And always, a population with test results is a biased sample. So I'm going to discuss this paper now that we published in International Journal of Epidemiology, using data from UK Biobank. This is a large biobank study of 500,000 people in the UK. And by biobank, I mean a large epidemiological study where we have biological material, a lot of baseline information, and linkage to electronic health record data. At the same time, we had the data of all the UK citizens who tested positive for COVID-19. And by linking these two, we have a subpopulation that has been tested among the UK Biobank participants. And we have the test results. Now here, the UK Biobank, we had the bias sample of people that wanted to take part in the study. So the study started before the pandemic, but yet we had people that were interested to join the study. So we had here a disproportionate amount of white, wealthy and healthy individuals. Here, again, it's a bias sample of people that were tested for COVID-19, especially last year at the beginning of the pandemic, where people were tested for COVID-19, whether if they were hospitalized or if they had severe symptoms. We didn't have the extensive rapid testing, which again is biased, but maybe to a different extent. We also had, the inaccuracy of the results, so we have false negatives and heterogeneity across tests, across different populations, different geographic locations, etc. So we came up with 4 different sets that we could compare. So we had tested versus not tested. So that would give us the probability of being tested in the population. And we had those tested positive versus not tested. And that could be a good comparison to study the risk factors of developing, COVID-19 disease, because we have tested for the disease versus not tested at all. We had those tested negative versus not tested, and we hypothesized that because they are tested, they had symptoms, so maybe they had something else. And those tested positive versus tested negative. We would expect that these positive versus population controls and those tested positive versus negative might have similar results, because both should be, the control population. And then I'm quickly presenting you the results, and I haven't gone deep into the methodology here, but these are univariate analysis, meaning that we look one risk factor at a time. And we have the 4 colors, which gives you these 4 comparisons. So the blue is the probability of being tested, the beige is the probability of being positive versus population controls. The green is tested negative, and the orange one is the probability of being tested positive given that you are tested. And you can see here the results. I don't think we find anything stunning. We find, first of all, these are the risk factors that we found to be significantly associated, but we tested much more for that, adjusting for multiple testing. And we have risk factors that have been previously associated with COVID-19 disease, like the ethnicity, the lower deprivation reflected by education or housing or income, and then either environmental risk factors here. Here, for example, we had environmental pollution, the PM10 and 2.5, all associated with all comparisons. Comorbidities, body mass index, smoking, etc. I'm going to reflect on those results, but then just to mention that we also did more sophisticated models where we use logistic lasso models to account for the correlation and the joint contribution across these covariates. And overall, I would say that here, you know, we can see that being younger, being a man, a non-white background, ever smoker, non-drinker, overweight, being on more than one medications, have lower socioeconomic status, and being exposed to environmental pollution, they all found to jointly contribute to a higher probability of being tested. And then models for the probability of being a confirmed or a suspected case selected fewer from those predictors, but consistent set of predictors. The same for models, the last models of testing positive conditional on being tested, where we had again 26 covariates, including again age, gender, ethnicity, educational attainment, obesity, and having had severe diseases like cardiovascular disease, and exposure to environmental exposure. So if I was summarizing these results, I would say that, you know, risk of being tested had to do with the conditions and the variables that I just mentioned. But then risk of testing positive had a lower set of predictors. That was male sex, lower educational attainment, non-white, and environmental exposure to pollution, air pollution. So here the health risk factors and comorbidities were not found to be associated with this. They were found to be associated with the risk of being tested, but not with the risk of testing positive. And maybe this has to do with the fact that those are associated with the risk of developing COVID symptoms and therefore being tested, rather than with being tested positive versus negative. So these factors probably have to do with symptomatic COVID-19 that is correlated with the probability of being given a test. And these results need to be interpreted by thinking that the positive tests are true cases, because we know we have very good sensitivity, but the negative tests could contain a proportion of cases, especially at the beginning, where a lot of these tests were given very late at hospital admission. And we know that the sensitivity of the test, when given at late stages of the disease, could decrease. So maybe the best comparison one could do is the tested positive versus the population controls, rather than those that tested negative, as the tested negative might be a contaminated sample, especially since they are tested if they had symptoms at the early stages of the pandemic. Yes. So overall, I wanted just to give you a brief, flavor on how useful this data could be to make predictions, to use the data to develop, decision analysis tools and decision tools for healthcare providers, for settings like vaccinations, etc. And how careful one has to be with the design, because these are all biased samples of different populations. They don't reflect, we know this from the everyday data we get from the number of cases each day. So the samples that we have are so biased, so one should be very careful on how to design the study and the comparisons in order to get valid results, because we saw here how much the result varied according to different comparisons. And how much need we are in this type of data for our population as well in Greece, where at the moment we don't have this access to, you know, all the testing data and the different demographics of the population and the geography to be able to make valid inferences. And I would stop here. We would be happy to have questions.
Thank you very much. Just a quick question at the end. You say we don't have the data. You mean as a country, or your group has no access to it yet?
I think as a country, or very few people might have it, but you know, we haven't. I think we haven't pulled together this data in a single database across different, you know, domains that maybe we have bits of different data.
Have you talked to people? I don't know who would be the ministry, or is there an effort? Is there a national effort?
Look, it is part of the Εμβληματική δράση. I don't know how to call it. In English, where Athena is also contributing. You know, there it is our aim to develop something like this. But you know, because at the beginning all the efforts were on to develop the testing, and now to do the genome bioinformatics for the mutations, I think that's now our next priority.
Good. Okay, good.
Okay, Dibara.
Any questions or comments?
Can I ask a question?
Faidon, please.
Yes. I would like to, by the way, very nice talk. Thanks for this. I would like to ask your opinion about, for instance, you know, the false positive extent of the tests we usually do, and if there is a way to systematically account for it in the model.
Yes, we can account for it. I'm less worried about the false positive. I think most of the cases are true cases. The problem is more with the negative ones, where some of the negatives could be cases, especially when the testing is late on the disease course. And on those that we don't have the data, so we don't know who has been exposed to the disease, and we only have some people tested, so it is very difficult to make this kind of inferences.
Okay, thank you.
Any other question?
Is there any kind of initiative at European level or global level where these kind of repositories that you presented for UK might be there? Okay, we might not have in Greece yet, but what about other countries? Do they share their data, or do they have a, let's say, they are willing to share or have this prospect in mind?
Yes, I mean, there are a lot of initiatives to, you know, use electronic healthcare data, especially for COVID-19. A lot of these data are difficult to be used because they are routine data from hospitals and require a lot of processing. Yet I think we become better and better in this, and we can use more and more of these tools. There are large collaborations now ongoing, and you know, even in the UK, there are a lot of groups trying to use different kinds of data. So the NHS digital data, all the primary care data, etc. And I think they would be very useful for now people studying long COVID, for example, where we want to see how the follow-up of people that have been infected, and what's their course and what's the follow-up we have in terms of hospitalizations or visits to clinical practice, etc.
And a more provocative question, from your experience, at least at national level, then the prediction models that you know, they are currently available, how effective do you think they are without having sufficient data possibly to use? I'm sure that colleagues here are much more, aware of how things should be if it is to have this kind of prediction model, and how many data one should have in hand for those to be credible.
Yes, I don't think at the moment we have any credible models. I mean, I talked about the Q risk, which is specific to the UK. And if one has to use it somewhere else, it needs to recalibrate it. So it needs to have the intercept of the other population that you are going to use it. I'm not aware of models that we are using, for example, to comment on that. I think we don't.
Okay, thank you.
Welcome.
Anybody else? We maybe have time for one last question. If not, I would like to ask you about the first part of your talk. So it seems that the issues are mainly methodological. Yeah. Is there any issue on data collection or on available data? I guess it's a bit related to the methodology, I mean, what people use, but is there any more technical issue that people do not have access to certain data or do not analyze part of it?
Yes, also that, that is, people also don't have access to big data sets. We saw that a lot of these studies were very small, only a few variables were collected. So, and a lot of the times you are constrained to the predictors that have been collected by, by chance a little bit. So there will be a lot of other things one would have wanted to include in a prediction model, but this information is not available a lot of the time. Or it is missing, and it's not missing at random. So the imputation is very difficult and often won't solve any problems.
Yeah. Okay. There is one more question by Haris Papageorgiou. Harry?
Yeah, hello. Thank you very much. Thank you for this very interesting talk. Regarding the systematic reviews, what I would like to say and ask possibly for a comment from you is about how to build these systematic reviews and how to analyze these systematic reviews. We are also working in this field. We've been working with automating in a way of how to automate the systematic reviews by applying AI and NLP techniques and in the whole process in an end-to-end fashion. So we first have a good, we have a database with all PubMed publications, and then we try we are using an information retrieval system to filter out what is relevant and what is not. And then we have a question answering system that actually poses questions in natural language and try to figure out where the pico elements are found in the publications. And by doing this, at the end of the day, we consolidate all the elements from the different publications related to the topic. So you mentioned that there is an AI tool in your workflow. Can you elaborate more on this?
Yes, it was an AI text mining tool that was used when we initially selected the abstracts and the titles to select a subsample of those that were more likely to be, relevant. That was, I think, a risky thing that we did because these tools are difficult they don't always work very well. We piloted it in terms of, we selected some studies to do it ourselves and the tools, and if you look at the supplement of the paper, it's been described in more detail.
Okay.
So it had good sensitivity, and we used it just to select the subsample. The prediction models have a very bad, how do you call it, keywords. So it's very difficult, and we don't have like, good algorithms in PubMed to select those studies.
So it's mostly a retrieval exercise. You pose a set of keywords to retrieve relevant documents from the PubMed search engine. Was it a PubMed? Yeah, so it is. So it's a retrieval. No, I'm talking about a different thing. I mean, yeah.
But that's very interesting, but we haven't done it. And I think, it's an area under developing, but we need it for the systematic reviews because, otherwise they take so much time that they come later than we need them.
Yeah, I know. Yeah. Thanks.
Thank you.
Okay. Thank you, Ioanna. We'll move on.
Thank you very much.
Thanks for being with us. Let's go on to our last and third session, which is focusing again on modeling the spread of COVID-19. So Pano, are you ready? So the speaker is Panos Macheras, Professor Panos Macheras. I think everybody knows him. He's very active within Athena and at different aspects. So he's heading the unit of Pharma Informatics. And today he's presenting results on modeling the worldwide COVID-19 spreading. So Pano, the floor is yours.
Can you hear me now?
Yes. Hello.
Okay. So, as you know, I'm not an epidemiologist. So I would like to describe my work on this field, which is really hot. And here is the outline of my talk. I will give some, aspects of what we call fractal reaction kinetics, which is the crux of the matter in my talk. I will also present some work we have done on Monte Carlo simulations in drug release, which is an interesting subject in my area of pharmaceutical sciences. There is an SMS message, actually, which activated my interest in the topic. I will present some data about the classical approach on the wall-mixed hypothesis susceptible infected model. And then I will describe our work on a fractal approach of dealing with the susceptible infected model, and I will conclude at the end. So this is the starting point of what we call today fractal reaction kinetics. Raoul Koppelman is a professor at the University of Michigan in Ann Arbor, the Department of Chemistry, and he published this article in 1988. A very nice coincidence, since College of Pharmacy and the chemistry department was next door. Unfortunately, I didn't know about Professor Koppelman at that time. And later on, about 2 or 3 years ago, later, in early 90s, I realized this very interesting, approach. And I realized that a professor from the University of Thessaloniki with the name Panos Argyrakis was a student of Raoul Koppelman. So I contacted him immediately when I started working in the area. And since then, we developed a very nice collaboration. Let me give you some the basics of fractal reaction kinetics. So this is more or less the law of mass action. When we have a bimolecular, say, reaction between A and B, the rate is expressed with this simple relationship, which says that there is a rate constant, and you can see A and B, the concentration of the two reactant species. So this is typical. Everybody knows this in elementary, say, chemistry. Since K is a constant, is independent of time. But Raoul Koppelman said that, look, in some cases, when we deal with diffusion-limited homogeneous reactions in three-dimensional systems, this K is proportional to the diffusion coefficient of the reactant species, but it is not proportional when we have, say, lower dimensions. At that time, it was very, say, fashionable to speak about fractal structures. Therefore, they use the expression fractal, say, processes, and that's why they call this exponent H here the fractal exponent, since K depends on time. This simple, say, difference makes a lot of difference in all, say, considerations in reactions. Here is the typical, say, figure, which indicates that when we deal with processes where the rate constant is really constant, non-dependent of time, you can see the parallel line on the top. But Raoul Koppelman used simulations and provided a clear description and a clear proof that we do have processes which are more or less time dependent. In this case, you can see that the exponent H is not 0, it's 0.32, and there is a clear view of the slope of the second, say, line. This is the fingerprint, more or less, of what we call fractal reaction kinetics. Is this important? Yes, it is. If you see the meaning of fractal kinetics, you can realize that you can have two vessels with the same concentration of A and B, and this can be different in terms of rate. There is a simple example in this slide when we can see that there were differences in the rate of the reactions, because there is the involvement of the exponent H in the process. This is a pictorial view. You see two vessels. You see 2, 4, 6, 7, say, reactant species on the left-hand side, and also on the right-hand side. However, on the right-hand side, there is a critical distance, which is smaller in comparison with the left-hand side. This makes the right-hand side to have a reaction rate much faster than the left-hand side. So two identical fractal containers with identical concentrations of reactive molecules, but with different instantaneous reaction rates. You can realize that the probability for instantaneous reaction is obviously higher in the container of the right. This was the message, more or less, of this, say, fractal reaction kinetics. Since then, applications of this, say, approach was made in many disciplines. We tried to see how this type of kinetics affects the drug release. Drug release is more or less the process where the drug is released from the formulation. Also, it's how the drug is dissolved in the solution of in the gastrointestinal tract, for example. So it's quite critical. You should have in mind that there are many, say, formulations which have a kind of controlled release, and they are very successful. So it's very interesting to know the kinetics of drug release. So we try to collaborate with Professor Gerakis in University of Thessaloniki, and we started doing Monte Carlo simulations in order to study in detail the kinetics. Here is more or less the description of what is a Monte Carlo simulation for those who are not familiar with this concept. In this particular case, we are interested about, say, processes which are stochastic, and we perform experiments in the computer, mimicking the experimental conditions as close as possible. So you can see here an example of a cylinder which has been created on the computer. And we have placed different, say, particles, drug particles. The cylinder has dimensions, and we follow should be a kind of animation here.
Πάνω, αν μπορείς να το κάνεις presentation mode, τότε θα δεις και το animation.
Okay, thank you. Okay, you can see more or less five snapshots. Here is the most, say, at the beginning, and then it progresses. So we can monitor the number of particles as a function of time. So we can have a profile, how the drug is released. Here we have an example of, say, of a Euclidean space. The diffusion follows the classical laws of diffusion. And the probability is moving in the different directions around. And as you can see, we published this work, and we found that the viable function can describe the phenomena. What is important to note here is that the viable function is this one. This was used for years and years in pharmaceutical sciences to describe the drug release and drug dissolution. M is more or less the amount of drug dissolved or released from the formulation. And the viable function has this peculiar structure where on the exponent there is, you can see the time and the power on it. So we understood that the viable function actually describes a process which is first order, but with a time-dependent rate coefficient. And as you can see here, our results confirm this. These are our Monte Carlo results. You can see the viable function, how nicely describes the data. And at the same time, you can see the fit of the power law. As you can see, it deviates beyond the 60 or 70%. And this power law was used for years and years in pharmaceutical sciences. So we proved that the viable function is able to describe the entire process from the very beginning to the end using Euclidean, say, space. But we applied the same methodology in fractal systems. Here is a percolation fractal. And as you can see, we follow again the number of particles released as a function of time. And we got very nice behavior again with the different, say, simulations associated with the release of drug from the system. The numbers correspond to lattice sizes we utilize in the simulation. So to be honest, we did a lot of work in the area. and we published several articles, and we were able to realize actually what is going on. And I would like to present this data from this paper, where we are now in a position to correlate the value of the exponent with the actual diffusion mechanism. Depending on the value B of the time exponent, you can see that you have, say, a kind of a complex mechanism. If B is higher than 1, you have a classical Fichtean approach, diffusion in normal Euclidean space, diffusion in fractal or disordered systems, depending on the value of this exponent. So I applied this methodology not only for modern Calor simulations in drug release, but to other, say, phenomena I'm not describing right now. For example, I applied that in pharmacokinetics. I introduced the term of extent of absorption rate constant, the basic, say, parameter describing the absorption of drugs from the gastrointestinal tract. I said that, look, we have an absorption rate coefficient implying fractal kinetics. This was actually adopted in the literature. Later on, we expanded the work of fractal kinetics to fractional calculus, and we had also very nice applications. But now I'd like to restrict myself to the more or less the influence of these results in my work related to COVID. I received an SMS message from my younger son Spiros, which is this say report from the Washington Post in March 14. It was a very nice video, actually, describing the basics of epidemiology, would say. The take-home message was that, look, if you try to isolate the infected people from the susceptible individuals, you can control the pandemic. There were some very nice say animations where they used some obstacles between the infected people and the susceptible people. I'm presenting here a snapshot of the early phase of the disease spreading. You can see the 3 red dots actually are the 3 infected individuals. Around are the susceptible people. As I told you, it was a lovely presentation with very nice animation. I'm not presenting this now, but later on, as you can realize, when you have the late phase of the disease spreading, the entire picture changes, and we have very few susceptible individuals, but we do have many infected. So when I saw this for the first time in March 1 year ago, immediately I realized that this can be described with fractal kinetics, since the topological constraints of our, say, simulations, Monte Carlo simulations, are quite close, what we call, say, social distancing today. So I started thinking and about, say, epidemiology. I didn't know everything at that time, and I'm now going to present some elements of the classical approaches in the susceptible infected model. In all cases, the classical model relies on a basic hypothesis that the system is well mixed. This is the basic, say, assumption when they work with classical models. So I learned what means R0. Everybody knows today. I saw some very nice, say, pictorial presentations in the internet from different, say, sources. You can see here now 0 for Ebola 2. You see that 2 becomes 4. In the second case, 4 becomes 16. These are different, say, generations. I noticed that, but to be honest, I did not like at all. Since first of all, I did not realize what means generation. Which is the time frame? Which is the time frame? Which is the duration between the two generations? So really, I did not like at all. When I could not say find a specific connection between generations and actual time. I read in the literature, I realized that this simple rho 0 is an extremely, say, complex concept. You can read in the literature that R0 must be estimated, reported, and applied with great caution. In fact, if you ask somebody today, which is R0 for COVID-19 pandemic, you cannot get a specific answer. You get peculiar, say, answers, or different answers, or values ranging. I also realized that they use RT, and this is a rather simple approach, since it's the real-time R0, but in actual calculations, it's related to R0. It gives a nice information, but is only interesting at this very moment. You cannot predict anything with RT. You know what is going on now or tomorrow. Not more than that. So again, this was, say, a negative characteristic. So when I got the SMS message, I called to my colleague, Dr. Kosmas Kosmidis in the University of Thessaloniki, and said to Kosmas, look, Kosma, here we have a very nice area to apply fractal kinetics. And in fact, we started thinking in this aspect since the well-mixed hypothesis cannot be applied to epidemiological models, since individuals, unlike molecules in a stirred solution, do not mix homogeneously. Most importantly, this is particularly so when preventive measures are applied and the individuals have this, say, social distancing approach. Here is the, say, connection between fractal kinetics and actual, say, pandemic. Here is an example with quite different, say, probability in the left-hand side versus the right-hand side. You can see these two infected people indicated with red in the left-hand side. They have 3 persons around them, and therefore the probability is 7 to 10. If you see the right-hand side, only one is close to 1 red person. So the probability is 1/10. This is a static, say, figure, and you can see that when we take into consideration the movement of the subjects, and if we have, say, a perfect mixing, we approach what we call the well-mixed hypothesis. This indicates that the well-mixed hypothesis is a special case, is in a case which is rather difficult to have in actual practice. So based on this idea, we work on this and we managed to have some very nice results published in last August in PLOS 1. And we started our theoretical work based on the classical approach of the two populations of susceptible and infected. With I and S, we present the two populations. We know that the sum of subjects of susceptible and infected is equal to 1. And this is the fundamental differential equation for the classical system. Lambda is the proportional to the probability of an infected individual to infect a healthy one, and B is the recovery rate. This is the starting point in epidemiology models. So we said, look, if the conditions are not homogeneous, we should take into account that kinetics is dependent on the space. That's why you can see in the denominator the h as an exponent of time. You realize that when h is equal to 0, this is the classical SI model, susceptible infected model. There is a limiting case when h is equal to 1. In this case, the rate of infection is inversely proportional to time. But most importantly, when h is higher than 0, is higher than 1, then we deal with fractal kinetics. And we believe that this number will be the result of measures taken to promote social distancing and public awareness. So, in order to study the system better, we use the cumulative fraction of infected individuals as a function of time. And we got the differential equation 3, which has a very nice, elegant solution given by equation 4. If you see the equation 4, you can realize that it's quite similar what we called previously variable function, since again, on the time, we have a time exponent again. What is also very interesting is the limiting behavior. When h is smaller than 1, the limiting value is 1. But when h is higher than 1, we have a specific, say, asymptotic limit. This is very important, and I will discuss this later on. You can see here the results of our study. Please note that these results were at the very, say, initial phase of the pandemic. Here we analyze data from the beginning up to April 4, 2020. You can see in the plots that I do have very nice fittings. And the values represent the value of h, the fractal exponent in different countries. At that time, we created a map. What is the value of h at the different countries? Here is a picture of 55 countries all around the world. In the article was published in August 2020, but the data was up to early April 2020. So we were very happy with these results, since for the first time we managed to analyze the data without any, say, assumption about a well-mixed model. And on top of that, we did not use any, say, assumptions associated with R0 or RT or whatever. So we thought that it's nice to expand this work, and we thought that it's nice to describe the first wave, the second wave, and probably all waves, trying to represent the change of the kinetics from the beginning up to the end. We call the kinetics herd kinetics when we don't know anything. Everybody moves around, freely move, people move freely all around. Then the government decides to do some, say, preventive measures, and people are reacting, trying to avoid each other. We call this period as fuzzy period. And later on, when actually lockdown is applied or strict preventive measures are applied, we call it fractal kinetics, since we establish, say, a new behavior of the people. And when the measures of the governments are relaxed, unfortunately, we come back again with a herd phase moving to a fuzzy phase and again to a fractal phase. These 6 words summarize the behavior of the people during the 2 waves of the pandemic. And I would like to present the data. Initially, we substitute the parameter lambda with this alpha to 1 minus h, since it helps us to present the equation 5, where alpha is a first order rate constant, which means that we can express in terms of half-life. If you can express a process with a half-life, you realize how long you can achieve, say, a certain percentage of the process. And as you know, the half-life is related to the rate constant, with this very well-known relationship with associated with ln2, which is very well known through the years for the radioactive decomposition of isotopes. What is interesting to note is that when we deal with a special case h is equal to 1, this means that we are at the limit when we move from a classical system to, say, a fractal system. When you solve the differential equation for h equal to 1, you come up with a power of time function, equation 6. So the beauty of fractal kinetics is that for specific values of the parameter h, we represent everything. For example, when we have h equal to 0, we do have what is very well known as classical kinetics, or the exponential model, as we call it. When h is equal to 1, we see what we call herd kinetics. People are freely moving, but we do not assume, say, a well-mixed system. But when h is higher than 1, we have a completely different behavior. See the plots. If you see the plots only under fractal kinetics conditions, we approach an asymptotic limit. Is what we expect in a in a pandemic. We expect to control it. We expect to force it to a steady state. You see that the two behaviors associated with the classical kinetics and herd kinetics are quite similar. Since, as you know, when we have a, say, an exponential function, we can have always relationships with a power function if you apply the Taylor approach. Therefore, playing with the variables, with the parameter values, we can have a similarity between the classical kinetics and the herd kinetics. Please note also the marks on the graphs, which indicate point of inflections, which are crucial, since when we record the cases, the infected cases, we can realize if we approach a point of inflection. Overall, this picture indicates the differences between a well-mixed system, H equal to 0, and a not well-mixed system, H equal to 1 and H higher than 1. Here is the same, say, plot, but depending on the values of the parameters we use, we can have different behaviors in the case H equal to 0 and H equal to 1. We can have, say, an approach of one curve to the other, depending on the values of the parameters. However, what is crucial is that only the fractal kinetics gives an asymptotic limit. Therefore, the case is H equal to 0 and H equal to 1 is more relevant what we call herd kinetics, which means that at the very end, the entire population will be infected. In contrast with this basic concept is the fractal approach. The fractal approach means that the system reaches a steady state. So we utilize these concepts, and as you can see on the top, on the left-hand side, we give a simulation where we present the two waves, the two pandemic waves. Initially, we start with the power law, H equal to 1. You can see subplot A at the right-hand side. Then we move to a fractal kinetic system where we have H equal to 4.5. Between these two phases, we think that there is, say, a fuzzy period, since people do not react spontaneously to move from the herd, say, phase to fractal phase. Then when we reach the steady state, usually governments relax the measures, as we noticed in all European countries and many other places. Then we start again to move to a new phase, starting from herd kinetics, fuzzy period, applications of lockdowns, and again a fractal kinetics period. So by applying this scheme, we were able to describe the different countries. Here you see the plots. We have the data. At the same time, we have the fittings. You can see the X mark indicating the decision about the lockdown. So the fuzzy period is always very close to the lockdown announcement. And we present simultaneously the data points and the fittings. I'm emphasizing this since are not simple, say, infected cases, cumulative infected cases, are actual fittings of equations 6 and 5, describing the herd kinetics and fractal kinetics respectively. So we have 4 countries, France, Greece, Italy, and Spain here. Here is a more, say, a better view of the results and fittings as well.
Pano, excuse me.
Yeah.
Are we getting close to the conclusions? Because the time is running out.
Okay, so I will speed up, and I need 3 minutes more. All right?
Okay.
Thank you very much indeed. So here are the data, the results of the fittings for the various phases: the initial herd, the fractal, again herd, again fractal. You can see the fittings are marvelous, very nice, very small coefficients of variations, very high regression coefficient. We also present some results from inflection points estimated and observed. We also have at the very end some predictions about the state. Here is the behavior of Brazil and Argentina. You can see here that we did not reach as yet the point of inflections. The data are up to December 2020. Again, I repeat that these are not data, are data with fittings of the equations. I'm simply quoting USA and Sweden. You see at the top, USA means that they never managed to approach a steady state. What does it mean? Because there are so many different, say, approaches in the different states, I think that simultaneously herd kinetics principles and fractal kinetics principles are applicable in all phases of the pandemic in US. While in Sweden, you can see that we have a kind of herd kinetics from the beginning up to September. Then we have, say, a point of inflection indicating a self-organization of the society, trying to apply social, say, distancing by their own, without any decision from the government. Here are data about the value of Vita at the beginning of the pandemic, trying to mimic what is going on in relation to R0. As you can see, tremendous variability. Also, we were able to calculate the time needed for reaching a herd immunity level. And as you can see, the plot indicates that Vita is the principal parameter, not the size of the country and the population of the country. You can have future work in the area, applying the susceptible infected recovered model in different, say, applications. Conclusion, it's more akin to real life and can be used for the analysis of all contagious airborne pandemics. I say that everything is, it's all about geometry in the microcosm and the macrocosm. The distance is critical. The stereochemistry is critical for those who understand steric hindrance and stereochemical factors in the reaction, as well as the electrostatic interactions between small molecules and proteins. Here is my acknowledgements slide for the 3 colleagues I collaborate with them. I would like to add that Dr. Rapsomanikis also, he offered a work in the computational area. Unfortunately, I did not know, but I'm obliged to present this since Dr. Rapsomanikis is in the Athena family. Before closing, it's the last say message I received from John Ioannidis yesterday about the number of people, researchers who have been involved in COVID-19. Thank you for your attention for those who are outside Athens. And here are the list of my questions. Thank you.
Okay. Thank you, Panos. Let's get to questions and comments.
Yes, please. I suspect that everything is clear.
I think Yannis has a question.
May I ask a question?
Yes, please.
Unfortunately, I could not attend from the beginning. I had other obligations, but I attended most of this talk and a little bit of the tail end of the talk by Ioanna Tzoulaki. On this one, Panos, this is great, and how good you can fit and actually predict what is happening. Can you associate some of your variables to actual policy actions that can then affect the curves in a way that we want?
Thank you very much for this question. If you can see number 2 here, I do believe that if we reanalyze the data of the non-pharmaceutical interventions, applying modeling work, we can realize the result. I mean, if we apply something in May and you see some results in September, and you have the modeling work and the parameters in May and compare them with what is going on in September, you have a starting point to get an idea how a non-pharmaceutical intervention in a society is expressed in terms of mathematics, in terms of the change of the parameters involved. See, this can give a quantitative measure about the results of the policy associated with non-pharmaceutical interventions. So, but we never try to do this. We don't have the people to do that. It requires an enormous work. Papers associated with the non-pharmaceutical interventions have, say, 15 or 20 people working in a very famous epidemiological, say, lab like Oxford, Cambridge, London, etc. And this is the starting point for me. I'm a person with 3 brilliant persons around me, but this requires a very big, say, group and people having all this data. But these are available only in centers like Koch in Germany, London, etc., etc. So yes, the answer is yes, that it's very nice to correlate the mathematics and the parameters associated with the models in different, say, time periods.
Yes.
And if I may have a follow-up, and you are saying that this study and this formulation and the parameters are independent of the pandemic. So we can apply it on other pandemics also.
Yeah, since the pandemics are airborne pandemics, not
Yes.
people
Yeah, understand.
try to apply a social distancing approach in order to avoid the contamination. So it's actually, yes, it can be applied to the analysis of all contagious airborne pandemics.
Thank you.
Good, thanks. Anybody else would like to comment or question? Personally, I found very interesting the technology transfer from chemical analysis, a chemical viewpoint to social distancing. You mentioned geometry is king. How about distance geometry? I know, you know, NMR and so on. Maybe it's important mostly in the micro level, mostly at the molecular level. And this is maybe something we can discuss with your group and my group. But do you think using distances as a geometric tool could also enhance your model of the pandemic spread?
This is quite clear. The social distancing is number one. It's clear. All the results, all the feedings, whatever I did indicate this, that the social distancing between the infected and the susceptible people, it's the number one factor. All of us, we know that there are so many other factors which are involved in this pandemic, but the number one factor is this one. That's why I presented the work of Dr. Rapsomanikis, since he analyzed with his work the different behavior of masks in COVID-19. As far as the distance in terms of calculations associated with NMR or the chemical approaches, yes, this is at the molecular level, and these are very, very important in terms of the interaction of the ligand with the protein in general. And the work they are doing in Cambridge is really excellent, and since they are able to resolve what is going on in the microscopic level. But I use this expression that it's all about geometry, since it's real. In microscopic level, you know all the details of the molecular structure and all the details of the van der Waals forces, the electrostatic profile around the molecule, and you can apply methodologies to find out the best, say, configuration of the ligand and the macromolecule for a perfect, say, match. And in real life, however, you have to wear your masks and be far away from the infected people. It's unfortunately so simple, but so difficult to apply in our life. And the results of this study is it's like a warning about the importance of the social distancing. Actually, the work on fractal kinetics is a hymn of the social distancing. You can see how the disease is spreading in the various countries. Looking at the values of the parameters in the story. Sorry. Okay.
Good. Okay. Good. That's good. Thank you very much. So I don't see any more comments. So thanks, Panos. Πιστεύω φτάνουμε στο τέλος της ημερίδας. Σας ευχαριστώ όλους. Είδαμε πολύ ενδιαφέροντα θέματα. Ήδη είπα, ανοίγοντας τη συνάντηση αυτή, ότι η μονάδα θέλει να μας μιλήσει ακόμα περισσότερο για τη δουλειά της και αυτό είναι κάτι που το χρειαζόμαστε. Είδα σήμερα μεγάλο ενδιαφέρον και πολλά θέματα που ακουμπάνε υπολογιστικά προβλήματα. Αν μου επιτρέπετε επιγραμματικά, είδα 3 μεγάλες κατηγορίες. Από την πρώτη ομιλία Υπάρχουν θέματα τεχνητής νοημοσύνης, μηχανικής μάθησης στο cryo-EM, αλλά τα οποία αναφέρθηκαν και στη δεύτερη ομιλία με την ανάλυση κειμένου και δημοσιεύσεων. Υπήρξε ένας μεγάλος άξονας εργασιών σε simulation και στην πρώτη ομιλία και στην τρίτη, το οποίο πιστεύει ενδιαφέρει κάποιους από εμάς. Και υπήρξε επίσης και μεγάλη συζήτηση, κυρίως στις δύο τελευταίες, για μοντελοποίηση, στατιστική και μοντελοποίηση φαινομένων. Οπότε πιστεύω ότι υπάρχει κόσμος που ενδιαφέρεται, που ήδη εργάζεται σε κάποια αυτά τα πράγματα. Θα δώσουμε λοιπόν ένα ραντεβού με τη μονάδα Φαρμαπλή μέσα στον Απρίλιο, σκεφτόμαστε, και πιστεύω θα είναι ενδιαφέρον για όλους. Σήμερα νομίζω πήγε πολύ καλά, εγώ έμαθα πολλά πράγματα. Ευχαριστώ τους ομιλητές και σας χαιρετώ. Σας ζητάω μόνο να ανοίξετε την κάμερά σας για να πούμε γεια.